Thanks very much for the introduction and thanks for the invitation to be here. What I'm going to talk about this morning, in fact, is enabling drug discovery with diverse lead generation platforms. And I'm going to talk a little bit about the developments we've made over the last few years in our platforms in this area. To introduce you very briefly to WuXi AppTech, we're a global service provider working today with well over 5,000 drug discovery organizations, drug discoverers, full stop, enabling them with all kinds of different technologies at pretty much every stage of the drug discovery process, from target identification, through validation work, through hit finding, lead optimization, through into preclinical safety testing, manufacturing, and clinical studies. And we do that across all therapeutic areas and all modalities. So we support not only small molecule drug discovery, which is largely the topic of this presentation, but we also support large molecules, antibodies, therapeutic proteins, peptides, and oligonucleotide discovery, manufacturing, and clinical development. That's a very large platform. I can't possibly speak to all of those elements over the next 15 minutes or so. So I would encourage you, if you have time, to pop along to our booth, which is B90, or take a look at our website, or catch up with me or my colleagues during the rest of the course of this conference. But what I'm going to focus down on over the next 10 to 15 minutes are some of the developments in small molecule drug discovery, and I'm going to focus in particular on affinity-based screening to find leads for the starting points for drug discovery. You may think that this is no longer such a critical activity. We have lots of advanced tools in this area now. We could think about using virtual screening, and if I get time, I may talk about that briefly towards the end. We can also talk about using AI and machine learning tools to identify small molecule ligands. But the fact is that across the industry, we still run a lot of early stage hit finding programs to look for new small molecules. And that's largely because we continually move into new target classes, new protein classes, for which there's very little pre-existing medicinal chemistry. And therefore, we need to find tools to modulate those proteins, to fully understand what their functions are, to understand whether they're actually tractable as targets for drug discovery, and ideally to identify molecules that we can develop and optimize as potential drug candidates. To perhaps indicate how this remains something of interest to people, if we look back over the last 20 years or so, this slide comments on the increased throughput that's available to us now for screening small molecule libraries. And we've seen this go up from around about 10,000 compounds a day in a high throughput screen, maybe 15 years or so, to today we're routinely screening tens or even hundreds of billions of small molecules in a high throughput screen on a daily basis to look for new chemistry as starting points. So that's the background, really. And I'm going to talk about those assay platforms that are agnostic of binding sites or indeed function. So I'm going to focus in particular on affinity-based assays. We're not looking necessarily for molecules that are functional, but they may be functional. But we're looking for molecules and screening processes that perhaps identify binding sites that we might not have predicted, identify binding sites that we can use to tag molecules for other processes, the obvious candidate being targeted protein degradation, but also mapping the surface of a novel protein to understand where the opportunities might lie. And one of the advantages of using affinity-based technologies is this lack of predestination, if you like, of binding site. We actually are looking at every part of the binding surface of a protein and finding opportunities for small molecule affinity. And we can use that in many different ways, not only as directly therapeutics, but also building into other processes, other more complex molecules. So this is a little bit of the headline. I'm going to talk about ASMS screening, so affinity selection by mass spectrometry. I'll talk about DNA-encoded libraries, and I'll talk about fragment screens. If I have a little bit of time at the end, I'll talk about virtual screening, but frankly, I'm not anticipating quite having time to do that. I'd like to reference this quote from Francis Arnold, 2018 Nobel laureate for chemistry, which essentially tells us that the way we design and set up these experiments is very, very critical to the quality of the data that we get out of them. Francis said, you get what you screen for, which is another way of saying, understand the experiment you're doing, understand the state of the protein that you're using to design these experiments and run these assays, understand not only the purity of the protein, of course, but also understand how it's folded. Is it correctly folded? Is it functional? Is it aggregated? Is it in the right state of phosphorylation or other post-translational modification to represent the process that you're really trying to examine in a screening activity? So a few words about ASMS. This is a sort of high-level cartoon of what this process looks like. We're starting out with a protein, a purified protein, and we're incubating collections of small molecules to try and understand which of those molecules bind to that protein. Now, this is a fairly intense activity. It requires milligrams or even tens of milligrams of protein, so it's not something to be undertaken lightly. We're also, across the industry, very focused on being conservative with protein for these assays. So the industry standard is to pool compounds at the initial affinity selection stage of these assays. So we'll typically be screening libraries of several 100,000 molecules in pools of perhaps 200 to 400 molecules that then, of course, we have to deconvolute at a later stage. But we're using mass spec to separate the protein that has small molecule ligand attached to it, and then denaturing to identify the small molecules that are then washed off that we can detect by mass spec. And by manipulating the protein concentration in the experiment, we get a very crude ranking of affinity of those large numbers of small molecules for the target protein after that deconvolution step that I mentioned, which largely involves going back with each pool of compounds and retesting the individual molecules. So what are the pros and cons a little bit of this type of approach? Well, it's very, very fast to set up. These experiments can be set up with protein in hand in only a few days. There's no bias towards binding site. And again, this is very, very important for us in these kind of assays to be completely agnostic of the potential binding site of small molecules. There are so many opportunities now to use molecules that bind away from a traditional functional site, and it could be binding an allosteric site or a site that we could not have predicted. But there are often ways in which we can use that information. It's scalable to very high throughputs. We're typically screening several 100,000 small molecules, but this is very scalable. We could easily be screening millions of small molecules. And it's broadly applicable across different target classes. It's limited to soluble targets, which is not an insignificant limitation, but beyond that, it's very relevant to all soluble proteins. To look at that workflow with some exemplification here, I mentioned we're typically screening about 300,000 small molecules. This is a screen we ran recently. We had about a 1% hit rate on the primary screen, so about 3,500 small molecules identified. We then took each of those molecules and tested them individually, which gave us about a 20% confirmation. So we're at about 560 confirmed small molecules. And one of the things you'll hear from me as we go through these different platforms is that we're very keen on using multiple orthogonal confirmatory assays as follow-up to each of these activities. In this particular case, we used a radioactive assay, so in this case, a binding assay. And we tested all those compounds at 2 concentrations and then took the more interesting molecules into a dose response curve and ended up with 20 compounds with an IC50 below that of the pre-existing reference compound. So a pretty successful and very rapid and economical activity to find completely new chemistry for the protein of interest. I'd like to give a few examples as we go through of how this has been applied in different ways across the industry. And this is an example for the very highly drugged by multiple modalities lipid target PCSK9. A company involved in this work, which was published last year, ran an ASMS screen of about 200,000 small molecules, found the ligand shown on the left, quickly derived a crystal structure. And this was a case where the molecule was not functional, but it was a binder. And they took that molecule and developed it into a molecule that used PROTAC, or targeted protein degradation, to knock down PCSK9. So an alternative modality, the starting point of which is a small molecule identified using an ASMS screen. I'll talk briefly now about DNA encoded libraries. I'm sure many of you are familiar with DNA encoded libraries. So this is a platform that allows us to agnostically screen very, very large libraries. And we make libraries typically of 10 to 20 or even more million small molecules, and then pull hundreds of those libraries to make very large collections. Typically, we're screening 50 or even 100 billion small molecules in a single affinity experiment. We have a number of different business models to offer that, to support that kind of activity. The libraries are very diverse. We include not only traditional small molecule libraries, but we also designed a number of covalent libraries, and we have peptide libraries in this pool as well. This is an idea of the workflow in an experiment in this space. So the key thing to know about DNA encoded libraries is given away by the name, really. Every small molecule has a tag. That tag is a string of DNA, and that DNA tells the story of the synthesis of that molecule. That gives us the mechanism by which we're going to understand which molecule we're looking at when we fish that molecule out of an affinity experiment. Now, of course, the diversity of DNA sequence means that we can individually encode many billions of molecules in this way. Chemistry is typically based on 2, 3, or sometimes 4 cycles of elaboration. And so the molecules tend to have DNA tags that represent that sequence of events. They allow us to decode each individual step, each chemistry, and each building block that was used to synthesize the molecule. That library, which, to reiterate, exists as a pool and it never exists as single entities, goes into an affinity selection experiment, very conservative with protein in this kind of assay, typically using a few hundred micrograms of protein per experiment. A couple of rounds of affinity selection and washing allows us to generate an enriched pool of molecules that are sticking to the protein of interest. We then go to sequencing to identify those molecules. So traditional next-generation sequencing to identify the molecules that are enriched through the affinity step. And ideally, those molecules are interesting from a medicinal chemistry point of view and go through to a validation, typically off-target DNA synthesis and orthogonal confirmation of activity. Now, that's sort of glossing over a huge amount of bioinformatics and cheminformatics. So really the rate-limiting step in these experiments is analyzing all the data that's collected, understanding what it means in terms of the efficacy of the full molecules. But the nature of the DNA tag means that we can interrogate that data at the level of individual building blocks as well. So we very quickly identify structure-activity relationships. We identify common building blocks or common chemistries that are leading to active molecules. So it's a very informative experiment. It's, again, agnostic of binding site. So we're mapping the entire surface of a target protein for binding interaction opportunities. To give an example here, and this is some work published by GSK a couple of years ago, and it's focused on combining hits from a couple of different sources. So the molecule in the middle here was identified using a DNA-encoded library screen. It's a ligand for RIP1. But GSK had also run a traditional high-throughput screen and identified the smaller molecule on the left-hand side. And medicinal chemistry looked at these two molecules and created a program around combining the information encoded in the two molecules that ultimately led to the identification of the clinical candidate on the far right-hand side of this slide. So very, very briefly, with respect to fragment-based drug discovery, again, agnostic, the binding site. This is a technology that's really been enabled by the creation and the throughput that's available with biophysical platforms, in particular, microscale thermophoresis and SPR. And it allows us to use very small molecules, typically molecules below 300 or even 250 Daltons, to identify discrete binding site pockets in proteins, which we can then predict. The orientation of the molecules in those pockets, sometimes assisted by computational tools, sometimes assisted by structural biology. And then we can think about ways in which we can link those molecules up to find molecules that are much more potent. So that's essentially the theory. Why this works is that we can screen a very small library, typically 3,000 or 4,000 fragments, but that library is representative of a very large amount of chemical space and represents, the vast majority of binding site opportunities in a protein. So this works very, very well. Again, it's very, very fast. Our workflow typically uses MST as the primary screen, but we can also use SPR. We will almost always go to an orthogonal platform like NanoDSF to confirm those activities. And then really the key question from a medicinal chemistry point of view is, are we going to be able to crystallize those molecules, or do we need to drive their optimization using an activity-led or a computational approach? Both of these are available, and as you can imagine, it's very much a good strategy if you're able to collect X-ray or cryo-EM or NMR data to enable the design and the combination of fragments to make more potent molecules. I'll just show one brief example here, and this is an internal program looking at the target STING, which I'm sure will be known to many of you. We ran a high throughput screen using the MST platform, had about an 8% hit rate of the fragments, and ran dose response curves on all those fragments on the right-hand side. Confirmed all of those fragments using multiple orthogonal techniques. In this case, I'm showing the SPR, NMR, and NanoDSF data, ultimately identifying a handful of molecules with binding activity around the 10 to 20 micromolar range, and then, of course, a distribution towards lower activity. But in aggregate, around 60 novel fragments for STING. And I'll wrap up this section with an example, again, from our own work. This is a piece of work targeting USP7, a deubiquitinase enzyme, that really looks at the next stage of that process. In this case, we took a fragment ligand for USP7 and were able to crystallize it and use that as a starting point for a rational drug design program. So this work was carried out by our site in Germany. So I will pause there, and I'll just run through to my last slide, since we're up against the clock, and I, as always, have been overambitious with my slides. So the key message really that I want to get over here is that there are many, many different small molecule hit finding strategies, and it's very important to think about this at the start to understand what your protein does, how you're going to try and interact with it, and to map that against the multitude of different opportunities for finding ligands that interact with that protein. So there are many different technologies. Those technologies will often uncover different chemotypes, so it's, it should not be assumed that if you run multiple screens using even the same chemistry against in different formats, then you will find the same chemistry. That tends not to be the case. These are highly parallelized experiments, so they're very fast, they're very economical, and our advice is usually to run multiple screens in parallel and really look at the aggregate of the chemistry that you identify. So I'll stop there. Thank you very much for your attention, and again, thank you for the invitation to speak today.